Hello, in this video I'm going to go over gas transport in the blood. So the two gases we discuss are carbon dioxide and oxygen. Carbon dioxide can be transported through the blood using 3 different mechanisms, while oxygen is transported through the blood using 2 different mechanisms. We're going to first take a look at carbon dioxide. So as I mentioned, there's 3 different ways we can carry carbon dioxide in the blood. About 20% of all of the carbon dioxide that goes into the blood actually goes into red blood cells and attaches to hemoglobin. So most of the time we think of hemoglobin as being necessary for oxygen transport, which is true. However, it can also transport, and it does also transport, carbon dioxide. When carbon dioxide is attached to hemoglobin, it's called carbaminohemoglobin. 10% of the carbon dioxide that enters into the blood gets dissolved right in the blood plasma itself. And the remaining 70% of the carbon dioxide gets converted into something called bicarbonate, and the bicarbonate is carried in the blood plasma. We are going to concentrate on that third mechanism, transporting bicarbonate in the blood plasma. This is kind of complicated. It doesn't make inherent sense when we think about it, but after we talk about it, I think you'll have a good understanding of this mechanism. When carbon dioxide enters into the bloodstream, some of it is dissolved in the plasma, some of it attaches to hemoglobin, but most of it is converted into bicarbonate. And this conversion process actually occurs inside of the red blood cells. What type of capillary will we see this happening in? Do you think this is happening in a systemic capillary or a pulmonary capillary? Go ahead and pause the video while you come up with your answer. If you said systemic capillary, you're correct. Carbon dioxide levels are very high in the tissues of our body, and the carbon dioxide will diffuse into our blood, and once the CO2 is in the blood, it will get into the red blood cells and then be converted into the bicarbonate. The blood leaving the systemic capillaries, heading back to the heart, is always said to be oxygen deficient and carbon dioxide rich. But most of that carbon dioxide is bicarbonate. Once that blood gets to the lungs, we can't exhale bicarbonate. It's not a gas. What has to happen is we have to convert the bicarbonate back into CO2 when the blood gets to the lungs. This whole entire process happens in reverse then, meaning inside the red blood cells, the bicarbonate is converted back into carbon dioxide, and then the carbon dioxide can diffuse out of the blood into the alveolar air. Right now you're probably thinking, why does this happen? This sounds overly complicated. Why is this even happening? Well, it turns out that carbon dioxide plays a major role with blood pH, as does the bicarbonate. We'll talk about this pH situation in more detail, but let's take a look at the role of bicarbonate as a buffer. When that bicarbonate is in the blood plasma, it acts as a major buffer in the body, and it buffers the blood against any major changes in pH. This is really our first line of defense against changes in pH in our body. That's how important this bicarbonate is. So if we have something happening in our metabolism or our urinary system or our respiratory system that's resulting in big changes in pH, most of the time the bicarbonate in our blood plasma is capable of buffering it. If the bicarbonate is not enough, we can then move to the respiratory system and the urinary system for assistance. But as I said, this bicarbonate is a really important buffer. Let's take a look at how we make bicarbonate exactly, like what's going on here. We know that carbon dioxide is converted into bicarbonate. What happens in the systemic capillaries is that carbon dioxide leaves the tissues of the body and then enters into the systemic blood. Some of that carbon dioxide will then move into the red blood cell. Inside of red blood cells, we have lots of hemoglobin, some of which will be attaching to the carbon dioxide. Also inside the red blood cells, we have an enzyme called carbonic anhydrase. And of course, enzymes are proteins that speed up chemical reactions, or catalyze chemical reactions, in other words. Carbonic anhydrase is going to speed up a reaction between carbon dioxide and water. Carbon dioxide and water will naturally react together without this enzyme. The enzyme just makes it happen faster. When carbon dioxide reacts with water, it forms carbonic acid. Carbonic acid is what we call a weak acid, meaning once it forms, if it's in water, it readily dissociates into hydrogen ions and bicarbonate. Right now, maybe there's a red alert going off inside your head because you hopefully are thinking, hydrogen ions, if we add hydrogen ions to a solution, that means our pH is going to go down, and that means our acidity is going to increase. Right? That could be problematic. Thank goodness for hemoglobin. Hemoglobin, like bicarbonate, is an excellent buffer. The hemoglobin inside of the red blood cells can grab on to those hydrogen ions that are being produced through this reaction and buffer them. Don't forget, a buffer is sort of like a closet where you can store hydrogen ions or remove them. It just depends upon what kind of pH you're trying to maintain. Quick overview, what we just talked about. CO2 gets into the red blood cell. CO2 reacts with water to form carbonic acid, and this reaction is catalyzed by carbonic anhydrase. The carbonic acid readily dissociates into hydrogen ions, which get buffered by hemoglobin, and then bicarbonate. And this is what we were trying to do, was to get to the bicarbonate, right? So now that we have this bicarbonate, we need to get it into the blood plasma, because that's where it acts as that excellent buffer, is inside the blood plasma. We already have an excellent buffer inside the red blood cells, hemoglobin. What we don't have is an excellent buffer inside the plasma yet, so we need to get that bicarbonate into the plasma. And that's a little bit complicated. The red blood cell has to use something called a chloride exchanger. So basically, this is a protein embedded in red blood cell membranes, and this protein will exchange chloride ions for bicarbonate ions. There are plenty of chloride ions in our blood plasma, and there's plenty of bicarbonate inside of our red blood cells. So basically, this protein is just going to exchange the one for the other. So the chloride ion will move into the red blood cell in exchange for the bicarbonate ion, which moves out. And now the bicarbonate is in our blood plasma. The bicarbonate will actually stay in the plasma until we get to the lungs. Once we get to the lungs, this whole entire process has to happen in reverse. But let's not go there yet. Let's take a look at this whole process of making bicarbonate in a more pictorial format. Those things that look like hard-boiled eggs are supposed to be just like normal cells of the body that are actively consuming oxygen and making carbon dioxide during the process of cellular respiration. So the carbon dioxide is going to move into the capillary and then move into the red blood cell within the capillary. Let's take a closer look at what's happening inside of this red blood cell. So now we have our big red blood cell, and outside the red blood cell, of course, would be our blood plasma, which I said is rich in chloride ions. That carbon dioxide, when it gets into the red blood cell, it will react with water, forming carbonic acid. And this reaction is catalyzed or sped up by the enzyme carbonic anhydrase. Once we have our carbonic acid, it just falls right apart. It's a very weak acid. So it falls apart, forming hydrogen ions and bicarbonate. The hydrogen ions, if they're not buffered, they will acidify our red blood cell and melt everything inside of it, or denature it. Thank goodness for the hemoglobin. The hemoglobin inside of the red blood cells is an excellent buffer, and it can grab onto those hydrogen ions so that the pH inside the red blood cell doesn't change. Now that we have our bicarbonate, the last thing we have to do is get the bicarbonate into the blood plasma where it can act as that excellent buffer. Embedded in the cell membrane of our red blood cells are proteins that act as chloride exchangers, exchanging the bicarbonate for the chloride ions. Now we have this blood plasma that's very rich in bicarbonate. That blood travels to the heart, goes to the right side of the heart, and then gets pumped to the lungs. In the lungs, we need to pick up oxygen and get rid of carbon dioxide. But the problem is most of the carbon dioxide isn't actually carbon dioxide right now. 70% of it is bicarbonate. So we have to reverse this whole entire process so that we can actually get rid of the carbon dioxide. When the blood is arriving in the pulmonary capillaries of the lungs, we do have some carbon dioxide in the blood plasma and some attached to hemoglobin. That's about 30% of our CO2. Those are the first CO2s to leave. Those are the first CO2s to diffuse out of the bloodstream and into the alveolar air. And so we get this drop in carbon dioxide in the blood plasma. And it turns out that this drop in carbon dioxide levels in the blood plasma is the key to triggering the reverse process with the bicarbonate. So once those CO2 levels drop in the blood plasma, what happens is we are going to exchange the bicarbonate for chloride ions again. In this case, we're moving chloride ions out of the red blood cell and moving the bicarbonate into the red blood cell. The hemoglobin, which was buffering the hydrogen ions, is now going to release the hydrogen ions. Those hydrogen ions can then recombine with the bicarbonate, reforming the carbonic acid. Due to the presence of carbonic anhydrase, rather than the carbonic acid just breaking down again into bicarbonate and hydrogen ions, the carbonic anhydrase ensures that the reaction will occur such that the carbonic acid gets converted back into carbon dioxide and water. Because don't forget, enzymes can work in the reverse or forward direction. They can work both ways. It just kind of depends upon the current environment that they're in. The carbonic anhydrase helps the carbonic acid turn back into carbon dioxide and water. Once we have the carbon dioxide, it can diffuse out of the red blood cell into the alveolar air, and then it can be exhaled. Let's take a look at this process in a more pictorial format. On screen here, we have a capillary with a red blood cell and bicarbonate in the plasma, and then on the side we have an alveolus. When this blood arrives at the lungs, any of the CO2 that's dissolved in the plasma or attached to hemoglobin will diffuse out of the capillary into the alveolar air, which will stimulate the process of converting the bicarbonate back into carbon dioxide. So assuming our carbon dioxide has diffused out of the bloodstream to the alveolus, we're going to go ahead and do the whole process of forming bicarbonate in reverse. First thing we have to do is exchange chloride ions for the bicarbonate, getting the bicarbonate into the red blood cell. Once we do that, the hemoglobin in the red blood cell gives up the hydrogen ions that it was storing, and the hydrogen ions will recombine with bicarbonate, forming carbonic acid. Carbonic anhydrase will then catalyze the reaction of turning the carbonic acid into water and carbon dioxide. Then the carbon dioxide will diffuse out of the blood into the alveolar air. Piece of cake. There is a direct connection between pH. There's a really important connection between pH and carbon dioxide levels. See if you can fill in these blanks. Carbon dioxide reacts with water to form blank, which readily dissociates into blank and blank ions. So go ahead and pause the video, see if you can fill in those blanks. And might as well try to fill in the next one too. pH is a measurement of blank. All right, let's see if you got them right. Carbon dioxide reacts with water to form carbonic acid, which readily dissociates into bicarbonate and hydrogen ions. pH is a measurement of hydrogen ions in a solution. And we know that as hydrogen ions increase in a solution, pH will decrease. So this could be problematic. When there's lots of carbon dioxide in a fluid, will this fluid have more or less hydrogen ions? And will the pH be neutral, acidic, or basic? Go ahead and pause the video and answer those two questions. Let's see if you got them right. Whenever we have higher levels of carbon dioxide in a fluid, we will have more hydrogen ions. And that's because the carbon dioxide is reacting with water to form the carbonic acid, which dissociates into the hydrogen ions and bicarbonate. Because of this, because of the addition of the hydrogen ions, that means our pH is going to drop, and a drop in pH means things become more acidic. And if you're thinking, well, aren't the buffers going to buffer this acidity? You're right. A lot of the buffers, whether it be hemoglobin or bicarbonate, will be buffering this acidity. But because of the overwhelming amounts of carbon dioxide, usually in our blood, we can't buffer all of it. And this is why the pH is different between arteries and veins in the systemic circuit. Because the arteries are carbon dioxide deficient, the pH tends to be a little bit higher, around 7.4. Because the veins have higher levels of carbon dioxide, they have more hydrogen ions, and their pH tends to be slightly lower, more acidic, at around 7.35.